In this video, we will discuss about how the C1GalT1 knockdown suppresses malignant behavior of PDAC. In the previous video, we have understood that when the researchers check the pancreatic cancer cell patients data, they are able to find out that where the C1GalT1 is high expressed, uh, the patient has low survival rate, or we can say that C1GalT1 has an inverse relationship with the survival rate. So here they did some in vitro experiments to check out whether this C1GalT1 knockdown caused suppression of the malignant behavior of PDAC or not. So they used different kind of cell lines and this HDP non-transformed cell lines. Here first they check the C1GalT1 expressions. At the short exposure they can see some of the expressions of the C1GalT1 in some cell lines, but in long exposure they see C1GalT1 are present in all the cell lines to some extent. Thereafter they knock down this C1GalT1 using 3 silencing C1GalT1 RNA that are represented here as SI-GalT1-1, SI-GalT1-2, SI-GalT1-3, and they can see that this SI-GalT1 shows is a decrease in the SI-GalT1 shows a decrease in the C1GalT1 expressions compared to the signals that they have seen in the panel A in almost all the cell lines. So once identifying that their silencing RNA against C1GalT1 is working, they then check 3 different properties: viabilities, migration and invasion of the pancreatic cancer cells. And what they are able to find out is that in case of viability, they can see that when the C1GalT1 has been knocked down, they see a decrease in the viability of the pancreatic cancer cells compared to the control. For HPAF2, HPAC, and this MIA PACA2, BXB33, BXPC3, PANC1, for all the 5 cell lines they have checked, they can see a decrease in the cell viability when the C1GalT1 is been knocked down, or decrease in the pancreatic cancer cells viability. Thereafter they check migrations of those pancreatic cancer cells, and they can see that migrations is also reduced for HPAF2, HPAC, MIA PACA2, BXPC3, but not for PANC1. And these bright field images are also shown here, which can be seen that HPAF2 density got reduced, and similar results they have seen for HPAC, MyPACA2, BXPC3, but not for PANC1. And when we did the invasions study, and they are able to see decrease in the invasions also in the HPAF2, HPAC, MyPACA3, MyPACA2, BXPC3, but not for the PANC1. So indicates that for the PANC1, the migrations and invasions does not get much affected by C1GalT1 knocked out, but for the other cell lines, this invasions and migrations will also get affected with the decrease in the C1GalT1. And thereafter they check in the HPAF2 and HPAC the expressions of TN using VVA FITC conjugated lactin. As the C1GalT1 has been knocked down, they are expecting the high TN structure formations, and they can see the more TN structure intensity in the case where they use SI-C1GalT1-3 knockdown for HPAF2 and also for this HPAC. So this indicates that the truncation of O-glycan occurs with a decrease in the C1GalT1 knockdown, and that results in high TN structure formations. And they also did their study with the C1GalT1 overexpressions that are shown in the supplemental figures, and there they have find out that some of the cell lines shows upregulations in the malignant behavior of the PDAC, but the results are not uniform. You can check out the supplemental results. So far in these figures they are able to make a point that most of the cells will lead to the suppression of the malignant behavior of the PDAC with decrease in the C1GalT1 expressions. And I hope you understood this basic point that this figures wants to convey. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.